Just your reminder that we do have exam number 2 in this class on Friday. Okay? So don't forget about a suspiciously few number of you have started your McGraw-Hill Connect assignments. Remember that chapters 5 through 10 are due on Friday. Not that you can't catch up yet, but if you wait to the last minute, you're going to be struggling big time. Okay? They take like 40 minutes to an hour each. And if you wait till 9 a.m. on Friday morning, you're not going to finish in time. Okay? So don't forget about those. You still have time, but make sure you complete those McGraw-Hill Connect assignments by Friday at 11 a.m. And also, if you're listening to this from home, I know most of the people in here come every day, but you do have to be in class to take your test. So we have an in-class exam on Friday. All right, and our exam for exam number 2 is very similar to exam number 1. It has the same basic layout, the same basic number of multiple choice true-false matching. It's going to look very similar. All right, you'll have another Scantron sheet just like I handed out last time. All right, any questions about the test or anything else? All right, so let's get started. Sorry that projector's still not working. I tried turning it on, but. All right, so we are going to pick back up right where we left off. We're going to be talking about mutations. Oh, I skipped a couple pages. We're going to be talking about mutations, right? And what a mutation is it's a change in the DNA of a cell or an organism, any organism. It can be a bacteria, an animal, a plant, anything. It's any kind of DNA change. The key to it being a mutation, or what we consider a mutation, is that it is a heritable change. What I mean by that is something that can be passed to the next generation. Okay, so it is very, very common for people to have changes to their DNA, to cells in their DNA. It happens almost every time you go out into the sun. You get sun damage. Sun damage can change the DNA in your skin. It can change, the UV light in the sun can change the DNA in your skin. However, your skin has nothing to do with how you reproduce. Okay? So those changes in the DNA to your skin are not going to be passed on to your future children because that is not an effect to your ovaries or your testes. It's on your skin. Okay? So a mutation is considered a heritable change to the DNA of an organism, a change that will be passed onto future generations, or it can be passed onto future generations. And a lot of times, genetic mutation is the driving force of evolution, because it's really important to remember that mutations are random. Okay, they happen because of random outside effects that we're not going to look into too much, but it can happen because of drugs. It can happen because of toxins. It can happen because of genetics. It can happen for a lot of different reasons. But those changes are random. And whether or not they get passed on to the future generation is also random, especially in organisms that sexually reproduce. But because we typically think of, when you hear a mutation, you typically think of mutation meaning something bad. And it's not. A lot of times mutations are neutral. They don't mean anything. Nothing happens. A lot of times mutations are good, or they make some kind of change that isn't good or bad, but it causes something to change. For example, rumor has it that the green color of eyes is a genetic mutation, one that got passed down over centuries and centuries and centuries, right? Green eyes have existed for hundreds of years. There are rumor, I don't know how much of this is true, that like curly hair in white people is a mutation. Okay, that is a change that just happened. It was a mutation that happened and it got passed down. The development of antibiotic resistance, which is arguably a really good thing for the bacteria, is a random event, and it's a good one. Okay, so lots of things can happen randomly, and that is what forces evolution. When mutations happen that provide something good, provides the organism with some kind of advantage over their fellow organism, right? A bird mutates to be able to live longer in cold environments. They're going to survive better when it's colder, and they're going to be able to reproduce better, right? Humans evolved. If you look at other organisms, when humans became upright, we evolved for our babies to have smaller heads because their heads would not fit through our tiny pelvises. Okay, so these are good things that allowed our species to evolve and change to fit the changing and evolving world. Mutations can be and often are very good, but mutations, again, they're random. They can be good, bad, or neutral. You never know what you're gonna get. So we are gonna talk about the different types of mutations, the categories of mutations, and how those mutations happen. Now, most of the time, like we kind of talked about briefly, is that a lot of times when a mutation changes the DNA, so when you have a normal gene, perfectly normal gene, normal DNA, that will give rise to a normal protein. But when you have a mutated gene, a gene that is changed or altered in some way, that can result in an abnormal protein or no protein at all. Now again, abnormal doesn't necessarily mean bad, but it can be. Abnormal could be the key to some new great, eye color, right? Or whatever else, but it can also be bad. But when you change the DNA, a lot of times you're changing the protein. You're changing the shape of the protein, you're altering its basic structure, and therefore you're changing its function. The lock and key design of that enzyme will no longer work. Okay, so there are 3 basic types of mutations. And the words should be pretty self-explanatory. Hopefully it's nothing too confusing. But the first type of mutation is called a substitution mutation. And in a substitution mutation, you're substituting. Imagine that. So a substitution mutation is replacing nucleotides with other nucleotides, with different ones, but it's always the same number. So if you're either replacing 2 nucleotides with 2 nucleotides, or 3 nucleotides with 3 nucleotides, or 10 nucleotides with 10 nucleotides, doesn't matter, it just has to be the same number. Or it can be 1 nucleotide with 1 nucleotide, as long as that's the same number. We have a picture of that down here, where you're taking this pink chunk and substituting it out for the green one. Okay, this is 3 bases, 1, 2, and 3, and you're substituting it for 3 other bases in the DNA. Okay? You're just swapping them, substituting. And then insertion and deletion, again, are exactly what they sound like. Insertion is adding any number of extra nucleotides. So this is an insertion right here. You're adding these yellow nucleotides. And again, this can be any number. It can be 1, 2, 3, 10, 50, 1,000. It could be any kind, any number of nucleotides. But you're just inserting them. You're adding them in. Insertion. A deletion is the deleting of any number of nucleotide bases. Okay, you're just erasing them. So in this case, we're erasing these pink ones. They're just going to be pulled right out of the DNA. Deleting them. And again, that can be any number. 1, 2, 1,000. Doesn't matter. Any number. Okay. Now, when it comes to mutations, we have, just like everything else, we have categories within categories. So of the substitution mutations, there are a few different types of substitution mutations. Now keep in mind, these are just the substitution. These are not insertion deletion. We'll talk a little bit more about those in a minute, but this is just substitution. And your page looks a little bit different than this one. You had something that looks like this underneath. It's not incorrect, it's just very confusing the way they have it written, because we didn't cover everything that you're supposed to cover when talking about this. We kind of skimmed it. So this is super confusing. So we're crossing off that top bit and just making that bottom bit bigger. Okay, so that's what I did here. So make sure you do that on your page as well. Put down those bases that are less confusing. Okay. All right, so what we're looking at here is this over here on the left is what we call the wild type DNA. Okay, and wild type just means normal. Okay, wild type is just scientist speak for normal. What you would expect to be in the wild. What most organisms of that species have as their DNA. Okay, and obviously we're just looking at a teeny tiny little chunk here to demonstrate what happens. Okay, so this is the normal DNA, and in the normal DNA we have TTC, right? And then the mRNA from that, T always binds to what? A. T always binds to A, and then C always binds to G. So our mRNA would be AAG. Now, if you go back to your genetic code, and you can go back and confirm if you'd like, but if you go to your genetic code and you look this mRNA up on the genetic code, you will find that it codes for this amino acid right here called lysine, or LYS. Okay, lysine. So that is the normal one. Okay, but there are a couple of different ways that we can mutate this. Okay, and so this first one is what we would call a silent mutation. And so what we've done here in this mutation is remember that the normal DNA is TTC. TTC. What we have mutated it to is TTT. That's different, right? We substituted that last C for a T instead. Okay? So if we substituted that last T, the DNA is now altered. We've mutated it. It's a substitution. The mRNA is going to read AAA. Because we changed the DNA, the mRNA will now be different. However, this is called a silent mutation because if you go look up AAA on the genetic code, it will also code for lysine. So even though the DNA and the mRNA are changed here, the protein will be unchanged. And so there will be no observable difference in this organism because their proteins are exactly the same way that they were before. That's why it's called a silent mutation. A change has been made to the DNA, but that doesn't show in the protein. It's silent. Okay. Now I'm going to skip the middle one just because I'd like to talk about it in a different order. But this last one over here, this is what we call a missense mutation. And in this case, our normal DNA, remember that we're going back to the normal one when we're comparing it. So the normal DNA is TTC. But in this case, we have swapped one letter, that middle one, from a T to a C. So we've made one change. Again here, we've substituted that second T for a C instead. So our DNA reads TCC. Okay? So when your DNA reads TCC, the mRNA will read AGG. Because T always binds to A, C always binds to G, and C always binds to G. AGG. And if you look up AGG on the genetic code, remember the genetic code is always meant for RNA, not DNA. So you're going to compare the RNA to the genetic code. If you look up AGG on the genetic code, it codes for the amino acid arginine. Okay? Arginine is different than lysine, right? That's a different amino acid. So the result of a missense mutation is one change in the DNA results in one change in the protein. We have changed what's supposed to be a lysine to what is now an arginine. We changed one amino acid. Okay? And that may or may not have an effect on the final protein. Sometimes one amino acid change doesn't really mean anything. Sometimes it means everything. We're going to talk about an example in a minute. We already talked about sickle cell anemia, but we'll talk about it again. So a missense mutation is changing, substituting a nucleotide for another one, which results in a change in the amino acid. A silent mutation again, is when you change the DNA, but the amino acid stays the same. And then our last type of mutation is called a nonsense mutation. And in this one, we're substituting that first letter. So instead of TTC, we have changed that first letter to ATC. And when you convert that to mRNA, A always binds with what in RNA? U. So it'll read UAG. And if you go look up UAG on the genetic code, it codes for a stop. Okay? So in the grand scheme of an entire protein, instead of substituting one amino acid for another, we have completely stopped translation. So changing one nucleotide has completely stopped translation. It codes for a stop codon, which means that translation will completely stop. So that means that the protein, whatever it was supposed to be, will be stopped much earlier than it's supposed to be. Now, there can be a difference in the severity of these mutations. If this mutation happens at the very end of a protein, it might not make that much of a difference. But if this protein happens, in the first part of the protein, then the protein is basically non-existent. Okay, so you'll have a completely abnormal, what we call truncated protein. It means it was stopped early. Okay, and so this is kind of a weird way of thinking about these things, but I like this example. Yeah?
So, depending on what letter you change, if you change the first one always, is it always going to be a stop?
No.
Okay.
No, it depends on what you're changing it to, and you're always going to have to look it up on the genetic code, because I don't expect you to have that memorized. But it is not, I think it just happened to be that case here, where the first one, changing the last one resulted in a silent or whatever. So it's not always dependent on which place you're changing. It's just dependent on what you're changing it to. Okay. All right, so this example here, I think is kind of a fun way of trying to conceptualize what a silent and a missense mutation would kind of be. And then also we're taking a second look at what an insertion mutation would do. So this is a sentence that I have written out, and I specifically have them written out in 3-letter words, just like you would read a codon, right? Codons are 3 bases long. So we have a sentence, the original, also known as the wild type. And when you read it, it says the cat ate the fat rat, okay? That's a sentence that exists. Sure. Now, in a silent mutation, if we changed this first letter here, what was originally a C, and we change it to a K, we changed the sentence. I changed a letter. But when you read that out loud, how would you read it? How would you read that second word out loud? Cat. It's the same thing. If you were reading that out loud, you'd still say the cat ate the fat rat. So even though I changed the spelling, I changed the DNA, the end result is still the same. The end result of me saying it out loud is still the same. However, in a missense mutation, if you change the C to a B, now you've completely changed the meaning. Because when I say this out loud, I'm talking about a bat eating a rat, not a cat. Okay, those are 2 different things. So changing one letter changed the sentence. Now, something interesting can happen that we haven't really talked about much when you do either, and this can be deletion as well. This example is specifically an insertion, but when you insert or delete bases, something can happen that is called a frame shift mutation. Okay, and that is because codons are always read in groups of 3. Okay, so I have specifically put up 3-letter words here. If I kept the spaces, like I said that they have to be 3-letter words, and I'm going to put the spaces between the words after every 3 letters, no matter what, which is how codons are read. If you insert or delete 1 letter, it screws up the whole sentence, right? Here we just added 1 C. But if I kept the spaces for the words the same, that doesn't mean anything now, right? I've screwed up the whole sentence. I didn't just change 1 letter to change 1 word. The entire sentence is screwed up. And that's because you're changing the reading frame of the DNA. Okay, so kind of back here, no, that doesn't work. But if you were looking at it in terms of DNA, and you can go back and try this yourself, but remember that DNA is always read in 3-letter blocks, okay, 3-letter codons. And if you insert or delete 1 letter, you shift the entire reading frame. Can I help you guys? You shift the entire reading frame. Sorry, there was like hovering back there, steering. You shift the entire reading frame. Okay? Now, these are some examples of sickle cell anemia, and I know we talked about sickle cell anemia before, but I wanted to bring it back by talking about, now that you have that kind of perspective about mutations, in a normal red blood cell, we have a protein called hemoglobin, and hemoglobin is responsible for carrying oxygen, right? We talked about this before. Hemoglobin carries oxygen throughout your body. So the wild type, right, the wild type hemoglobin DNA, this one small part of it is supposed to read CTC, which results in an mRNA that says GAG, which results in the amino acid glutamine, or GLU. Okay? But in sickle cell anemia, there is a 1 amino acid change. Instead of having a T there, it is now an A, which changes the mRNA at 1 spot. It is now GUG, and if you look up GUG on the genetic code, it codes for valine. So this is, what type of mutation is this? Missense. I think I heard it. This is a missense mutation. We substituted 1 base for another, and it resulted in a change of the amino acid. Change from 1 amino acid to another. That is a missense mutation. And this missense mutation, again, I know we've talked about this, but this one change, changing one nucleotide base to another in the DNA, causes the change of 1 amino acid, which causes the protein hemoglobin to be completely misshapen. It changes the entire shape of this person's red blood cells and affects them in their entire lives. Okay, 1 amino acid change, 1 nucleotide change results in a very severe illness that somebody will be fighting their entire life. And this is a genetic illness, so it is passed down from person to person. So this is what a sickled red blood cell looks like. It does not carry oxygen well. It gets literally stuck in person's veins. It causes incredible pain because those weird shaped blood cells get caught in your joints and your capillaries, and it hurts. It can cause blood clots. It can cause organ failure because those blood clots get caught in organs. Most people who have sickle cell anemia have to have hundreds of transfusions throughout their lives in order to stay alive. And all of that is true because of 1 single nucleotide. Okay, so 1 nucleotide change can be incredibly important. Just because it's 1 doesn't mean it's not a big deal. Now, something that's also kind of interesting, and this is just a side note, not something you necessarily have to remember, but does anybody know why, or I guess first of all, does anybody know what population of people sickle cell anemia is most common in? Yeah, it's most common in African Americans. Or people of African descent, not necessarily African Americans. It's most common in people who are of African descent. Now, does anybody know why? This is kind of a cool piece of evolution. I mean, it's not great for the people that have it. So sickle cell anemia, having sickle cell anemia actually provides protection against malaria. So people who have sickle cell anemia are much, much, much less likely to contract malaria. And so malaria is most common on the African continent. So people who live on the African continent were more likely to get malaria and were therefore more likely to get this mutation. Which is just an interesting bit of evolution. Because if the people who had this didn't get malaria, they were more likely to survive to be able to reproduce again. And they passed this on to their children. So that is how mutations can work both good and bad, right? Sickle cell anemia is objectively a bad thing. Having this disease is terrible. But if you lived in a country where malaria was common, it would be considered a benefit. So it's just kind of proof, and it's an example of how things aren't always as simple as we want them to be, that this is always a bad thing or always a good thing. That's very rarely true. Okay. So now that we've talked about all that mutations and all that stuff, I want to talk a little bit about how we actually test for DNA, because a lot of, and I've kind of hinted at this, we've touched on it a bit, but DNA testing or testing for organisms based on their DNA and things like that is a very, I don't know what the right word other than like up and coming. It's becoming a very popular thing to do. As the technology gets better, it becomes cheaper and more possible to do on an everyday basis. It gets more and more sensitive. The kits get more and more reliable. And genetic and DNA testing has obviously existed for a long time. You could always get genetically tested. But even like something that you guys might not even realize is kind of crazy is when I was a kid, sending off your DNA to be tested was like, nobody did that. That was not a thing that existed. Okay, I grew up, I was born in 1987, which sounds like a million years ago, but I was born in 1987. I grew up in the 90s and early 2000s, and sending off a tube of your spit to be tested for DNA is absolutely insane. If you would have told me that when I was 12 years old, I would have thought you were crazy. People didn't do that. And now, when even you can send off your DNA for $100 to 23andMe, and you get an entire ancestry report about people you are related to 16 generations back. And like another thing, when you are, and I don't know if any of you have ever been pregnant, but if you've ever been pregnant or heard of this, it's entirely possible now to have your blood drawn at 10 weeks pregnant, and they can test your baby. So you have your blood drawn, and they can test your baby's DNA, because your baby's DNA floats around in your own bloodstream. You can test your baby's DNA for DNA abnormalities. You can test them for the sex. So that's how you can find out what sex a baby you're having at 10 weeks pregnant, which is wild. A 10-week-old fetus is the size of like a peanut, but we can know the sex of the baby, all kinds of crazy stuff. And it costs like $150, right? It's a really simple thing. So genetic testing has come a long way in a very short amount of time. And it's only getting bigger and bigger and bigger and more common. So as you guys move forward in your careers, it is only going to be a bigger thing. And more testing is going to rely on genetics and DNA. So our first major procedure that was developed in DNA and genetic testing is called polymerase chain reaction. And it's arguably the most important discovery in genetic testing to date, because it's the basis of everything else, right? It was the first thing that was developed, and everything that came after it is developed on this one procedure. So again, arguably the most important. And what polymerase chain reaction does is it's a procedure developed to replicate DNA outside of cells, or basically in a tube. And that is what the word in vitro means. In vitro basically means in a tube. The opposite of in vitro, if you're curious, you may see it around in other classes, is in vivo, which means like in your body or in your cells. Okay, so in vivo means in your cells, but in vitro means outside of your body, in a tube. And this allows us to take a tiny bit of DNA from a swab or a cup of your spit, a tube of your spit, I guess is a better way to say that, or a little bit of blood from a mother. They can extract the baby's DNA from a mother's DNA. Or a tiny bit of DNA left in a bone from 2,500 years ago. All kinds of different stuff. You take one little tiny piece of DNA and you replicate it in a tube so that you have millions of copies. You are never going to run out of that DNA. Have millions and millions and millions of copies. And what that allows us to do is allows us to test for the presence of specific genes. And it allows us to pick apart DNA samples based on, if we're testing somebody for viral DNA, right? I want to test if somebody has the flu or COVID. Okay, well, I stick a swab up their nose, that's certainly going to pick up some of their DNA, right? You're going to get some of your own cells in there. I don't care about the human DNA, I just want the viral DNA. Well, you can design a PCR procedure that selects for the DNA that you want. Okay, this can be used in hundreds of ways. It's used all the time in like sexual assault cases where a female has been sexually assaulted by a male and there is like semen left behind or skin. It doesn't even have to be semen, skin, hair, anything else. And they select for DNA that has a Y chromosome because they know that she was assaulted by a man. Okay, she's not going to have any Y chromosomes. She's got X chromosomes. So it can be used in forensics, it can be used in genetic testing, it can be used in all kinds of different ways. And probably one of my favorite stories about polymerase chain reaction is the guy that discovered PCR. Again, arguably one of the most important discoveries made in modern scientific history. His name was Kary Mullis. And Kary Mullis was a scientist, and he came up, does anybody know where he came up with this idea? It's kind of a famous story. You may have heard it, you may have not. Kary Mullis was high driving down Pacific Coast Highway in California. He was doing LSD, he was high as a kite, driving down the highway, looking out at the ocean, and it just came to him out of nowhere. And now he's a billionaire and has probably saved billions and billions and billions of lives. Right? It's arguably one of the most important procedures developed in genetic testing ever. And he came up with it when he was dropping acid. I just think it's fun. Okay, so maybe doing acid's a good thing sometimes. I don't know. Who am I to say? I can't say I've ever done it, but who knows? Maybe it's the key. All right, so with viral testing, we're specifically going to focus, like I said, polymerase chain reaction is a procedure that can be used in lots of ways. We talked about how it can be used in forensics and all kinds of stuff. But one of the ways that's really important and really growing currently and very relevant to your future careers is viral testing. And there are two major ways of testing viruses in a lab. Because we talked about this briefly when we talked about viruses, that viruses are very hard to grow. Okay, the only way to grow a virus is to grow it inside of a cell that it is able to infect. So in order to grow a virus, you have to grow the cells first. That can be keeping human cells alive. It could be having chicken eggs, all kinds of stuff. But keeping alive viruses is very difficult. In contrast to keeping alive bacteria. You guys have all done it if you've been in lab. You swipe some bacteria on an agar plate and leave it at room temperature, the bacteria is going to grow. Okay? But viruses are much more difficult. They require a lot of babysitting and a lot of very intensive work. So a lot of times when we are testing people for the flu or COVID, any of those viruses, culturing them and trying to grow those viruses is not really a viable option. It takes a lot of manpower, takes a lot of energy, and it takes a lot of money. And it takes a lot of time. Growing viruses can take weeks. How useful is it going to be if you go to the doctor and you're like, I think I have the flu, and he's like, okay, well, we'll do a test and we'll tell you in 2 weeks. Your flu is gone in 2 weeks. That's useless. I need to know if I have the flu today, please. Okay, because they need to know if they need to give you medicine or how to take care of you. You need to know that information now. Culturing is not going to help that. So in recent times, there are 2 major ways of testing for viruses in the lab. The first one is called antigen testing. And what antigen testing is, and we'll talk more about what an antigen is later, but an antigen is mostly just something on the surface of a cell. It can be a protein or a sugar or a fat. It's just something that's on the surface. But what antigen testing is testing for is it tests for the viral cells or the viral particles themselves. So we're testing for some kind of sugar or some kind of fat or some kind of protein that is on the surface of that particular virus. So if we're looking for the flu virus, we're testing for a sugar that's on the outside of this particular flu virus. Okay? And antigen testing is very similar to, antigen testing is what you do at home, right? These are like the home tests where, right, you stick the swab up your nose and then you stick it in the tube and then you put a couple of drops on the little card and then it goes across, and if there's 2 lines, you have the flu. If there's 1 line, it's negative. Okay, what that is doing is testing for those antigens. Okay, now antigen testing is incredibly useful, but it is not very sensitive. Because what this test is depending on is that in that sample of you stuck this swab up your nose, that there is enough virus in there for the test to pick up the presence of the virus. Now, this is really, really, really useful if you're at home and you are in the middle of an infection, right? Like you have a fever, you have snot pouring out of your nose, you feel terrible, and you're like, I need to figure out if this is COVID, flu, or just a cold. Right? You're in the middle of an infection. When you're in the middle of an infection like that, the number of flu viruses that you have coming out of your face is by the million, right? There are flu viruses everywhere. So this is really great. If you're in the middle of an infection, it's cheap, it's fast, right? We've all done them at home. They take, 10 minutes? They're generally pretty cheap. You get them for like $10 at Walgreens. Okay, so for antigen testing, as you have over here, it takes minutes, very, very quick. It's cheap. It costs a little bit amount of money, but it's not very sensitive. It misses infections with low virus levels. So again, if you are in the middle of an infection, an antigen test is a great solution because you have plenty of virus to go around, and it's cheap and easy. And that's why we all keep them at home, or that's why they suggest you keep them at home, because it tells you if you have COVID or the flu very quickly, very cheaply. You don't have to go to the doctor and pay for a doctor visit. However, PCR testing, what PCR testing is testing for, or nucleic acid testing, is it uses PCR to amplify the number of DNA strands, right? So that's what PCR does. It increases, it duplicates the DNA in a tube. So in PCR testing, what we're doing is sticking a swab up your nose and then putting it in a tube and then putting it on an instrument that will do PCR on it. And it is going to selectively amplify that viral DNA in that sample. PCR testing will pick up an infection if you have 1 viral particle. If there is one, not even 1 viral particle, if there's 1 piece of DNA in that sample, it will amplify millions of times and it will test positive. Okay? And this does require special machinery, not just to do the PCR, but to do the analysis of the PCR, because you can't just put it on a card and look for 2 lines, right? It's much more complicated than that. So PCR testing usually takes hours, not usually days. PCR testing usually takes hours, but it costs a significantly higher amount of money, and it has to be done at a hospital or doctor's office that has the capability. Right? They have to have those instruments. Those instruments require a lot of maintenance. You have to have somebody that knows how to run them. It's not something you can just do easily. It's a very technical process. It requires a lot of technical ability. So it costs lots of money. It takes a lot longer. However, it is incredibly sensitive. Incredibly sensitive. Like I said, it will pick up an infection if you have 1 piece of viral DNA hanging out in that sample. So PCR testing is much more useful in situations where you've either been exposed to something and you are curious about whether you've picked it up, or you want to know, I guess it will also stay positive for much longer after you're done being sick. So yeah, it'll be positive much earlier than an antigen test. This is why we're getting to the point now. I used to be able to talk about COVID and everybody had been in college when that happened, but we're getting to the point now, you guys were in middle school, right? When COVID happened. And so you might not have been aware of as much about what was going on, but during the peak of COVID, there was a time that they wouldn't let you on a plane unless you had a piece of paper that had a PCR test result within the last 24 hours. And that was to prove, you don't have any symptoms. I don't have any symptoms. But if you were about to start showing symptoms, a PCR test would be positive. Okay? And so, again, there was a time where to get on a plane at the airport, you had to present a PCR test signed by a doctor within the last 24 hours to be allowed on a plane. Because PCR tests will show the presence of a virus much sooner than an antigen test. An antigen test is great if you're already sick. A PCR test is great if you're not quite sick yet. Okay. But again, the PCR tests are great and they're incredibly sensitive, but they cost a lot of money. And at the current point in our technology, a lot of insurance companies don't cover them because they don't think they're necessary. They don't think they're necessary, so they won't cover them. And so PCR tests can cost $600 or $700 per test, which is a lot of money. Not to mention that you had to schedule a doctor's visit, so you got to pay a copay on top of that and all that fun stuff. But I think in the next, probably won't even take that long, this technology is going to get more common. There's going to be, right now there's 2 companies that make the instruments for this. There's going to be other companies that come up with their own instruments, and therefore the supply and demand is going to go down. Things are going to get cheaper. I think in your guys' careers, genetic testing and viral testing through PCR is going to become much, much, much more common for all kinds of things, not just viruses. Okay. All right, so that's the end of that chapter. We are going to move on to chapter 9 today. So I'll give you a second to pull that up. Okay. All right, so chapter 9 is a chapter that focuses on, we've talked about we spent the last couple of chapters talking about how microbes grow, the types of environments that they need to grow. We talked about temperature requirements and oxygen requirements and all of that fun stuff. Well, now in this chapter, we're going to talk about how we as humans use that knowledge, use those ideals of how microbes grow and use that to our advantage to kill them or to control them. Okay, and controlling, this is just a funny little comic, but controlling microorganisms and controlling the growth of potentially harmful microbes is ingrained in almost all aspects of our lives. It's something that we do constantly, even if you don't think about it. Again, we've talked about brushing your teeth, right? Brushing your teeth is controlling the possibly pathogenic microbes that grow inside your mouth. We wash our hands after we go to the bathroom. We wash our hands after we touch something gross. We do that specifically after we go to the bathroom because there's a lot of bacteria in our colons and a lot of bacteria in the vaginal tract. And we are trying to prevent that from getting around, being spread by our hands. So we wash our hands after we go to the bathroom. We cook our food. We keep our food in refrigerators to prevent it from going bad. Most of us drink pasteurized milk products. The things you personally do all the time to control microbial growth, right? If you wear makeup, you clean your makeup brushes, you wash your silverware, you wash your dishes. All of these things, all of these little things are ways of controlling microbial growth. Even things like not being around sick people, staying home if you have a fever, right? That's another way of controlling the way that microbes grow. And so we're gonna look into it a little bit more, these are things that we kind of take advantage, but we are going to talk about how those different things work, why they work, why we do them, all that kind of fun stuff. Now, something that I think is important to mention, and I think I've talked about it before, but it bears repeating, is that we are never, nor should we ever try, but we are never going to kill all the bacteria. They're always going to outsmart us. There's always going to be more of them than there are of us. And we shouldn't. They do a lot of great things for us. If you killed off all the bacteria in your colon, you would be miserable. Okay? You might survive, but you'd be miserable. You shouldn't try to kill all the bacteria, and you never will. Most of the time, when we are talking about controlling microbial growth, we are talking about doing our best to limit the number of potential pathogens getting into a place where they can cause infection. And again, we're never going to fully succeed in that. It's not a reality that we are ever going to live in that we don't ever get sick. Okay, that's just not going to happen. But there are ways that you can limit the possibilities, right? Keeping food in refrigerators, cooking food, things like that, limit the amount of potential exposures, I guess is the best way to say it. Okay, so these are our basic types. There's 4 basic types of antimicrobial control, and we are going to talk about these in more specific in the rest of this chapter, but we do have to we have to do a little bit of vocabulary, which I know isn't the funnest, but we do have to talk about these specific things. Now, these words in general, you've probably heard of at least a couple of them, if not sterilization and disinfection, maybe some of the others. But in general, in the world, a lot of people use these words incorrectly. Okay, so you may use the word disinfect something or sterilize something. You may say that the dishwasher sterilizes your dishes. It doesn't. You may say that boiling baby bottles sterilizes baby bottles. It doesn't. Okay. It kills a lot of the bacteria, but it doesn't sterilize. And so in this class, it's important to have precise verbiage, okay? You have to use the correct word at the correct time. They mean specific things, and in this class, you have to remember what they mean, even if you have heard in the past. So try to not bring, again, we talk about sterilizing baby bottles all the time. People don't sterilize baby bottles in their house. It's not possible. The only way to sterilize something is to use an autoclave, okay? And you don't have autoclaves in your house. I know you don't. Okay, but that's a word, right? That's a thing that people say. I'm going to sterilize the baby bottles. You're not sterilizing them, you're disinfecting them. Okay, so it's really important in this class to have precise language and to know exactly what these words mean, even if they don't line up with some of the things that you have probably said before. And I'm guilty of it too. I still say sterilized baby bottles, even though I know in my microbiology brain that that's not true, okay? But just keep in mind for this test and this part of this class, you have to get the words right. So sterilization is a process that destroys all viable microorganisms. Viable means capable of reproducing. Okay. And this specifically includes endospores. So we talked about endospores in the last section. Endospores are a protective structure or a defensive structure that some bacteria are capable of creating that basically makes them go dormant. And I know we talked about this before, that endospores are incredibly difficult to kill. That's the point, right? That's why bacteria make them. They make them so that they can survive in incredibly harsh environments. So you are only considered sterilized, something is only considered sterilized if it also kills endospores and viruses. There are some viruses that are very difficult to kill. So the major examples of things that are capable of sterilization, we're going to talk more about this in a little while, but using an autoclave specifically, boiling is not enough to sterilize. Boiling is not going to kill endospores. Endospores are incredibly capable of surviving boiling water. They can survive UV radiation. They can survive soap, Lysol, things like that. There are very few things that are capable of killing endospores. So sterilization, again, is a process that destroys all viable microorganisms, including all endospores and viruses. Disinfection is a process or agent that destroys all vegetative pathogens. So vegetative pathogens are ones that are currently alive or metabolizing, not necessarily things that are capable of reproducing, because endospores are not alive. They're dormant. They're not currently metabolizing. They're like little, I kind of think of them as like seeds, right? They're like this dormant, you wouldn't call a seed a living plant because it's not. A seed is a dormant little structure that when you put it in the ground and water it, it will become a plant. But a seed is not a plant. Just like an endospore is not a vegetative cell. Okay, it's not currently alive. So disinfection is killing basically everything that's alive, but not necessarily killing bacterial endospores. And there are a couple of examples of this. Again, we'll talk more about this later, but things like bleach and iodine, heat, like boiling, not an autoclave, but like regular boiling will disinfect. Okay, so this is mostly what we're doing at home. At home, I can't really think of a situation where in your own house, you're actually sterilizing something. There might be some examples, but for the most part, you're disinfecting. Decontamination or sanitization is a cleansing technique that physically removes microorganisms to a level that is safe for use, okay, which is a little bit confusing, but something I don't think a lot of people realize is that soap doesn't actually kill bacteria. Soap just removes it. It just removes it from your skin. A lot of detergents and dishwashers are not actually killing all of the bacteria. They're not even necessarily disinfecting it. They're just removing the microorganisms to a level that is safe for us to use them. And that level is going to depend on what it's being used for. Decontamination / sanitization is a word that's usually used for some kind of public product. So this is like going to a restaurant, like restaurant kitchens have sanitization standards. They have levels at which they have to clean things because it's like a public use thing, right? People are gonna come in and eat the food. So it has to be sanitized to a particular level. It's also used for a lot of like products that are sold, right? Most fruits and vegetables and things like that are 100% going to have bacteria on them. but before they're sold, they're usually sanitized to a particular level. So anything that is going to be sold or used by the public bathrooms and things like that have particular sanitization requirements. So sanitization is usually a word that is reserved for like public use, public health kind of stuff. Okay, and then antisepsis and degermination is a process that reduces the number of microbes on human skin. So antisepsis is a lot like disinfection, but it's particularly on human skin. Okay? So when you're talking about using hand sanitizer or washing your hands, surgical hand scrubs, any of that stuff, that's not disinfection or sanitization. It is antisepsis because it's on human skin. Because you're also, no matter how much soap you use, in reality, you're never going to completely remove all the bacteria from your skin. Okay. Right, so I know that all seems a little pedantic and silly, but it is important to know the correct words that go with each thing. Okay, and then this next chart looks really scary, and you don't necessarily have to memorize this chart, but this chart is kind of I can't think of the word. It's kind of an overview chart, or a summary chart of the rest of the stuff we're going to talk about. Okay, so I think that this chart is helpful to revisit once we've talked about the rest of it, but I wanted to put it here. And this talks about, of the microbial control methods that we have 3 general categories. We have physical agents, chemical agents, and then mechanical removal methods, which we're going to talk about more. And then within those, we have different types, and it tells you whether or not these types sterilize or disinfect. And that's going to be important. Okay. And like I said, we're going to go through all of this individually, but this chart will be a helpful overview, I guess, summary. Okay. All right, so when you're choosing a method of control, how do you choose, right? We have all of these different methods. Why is it that we brush our teeth, but we wash our hands with soap? Why don't we brush our hands or wash our mouths with soap? Right? And the reason for that, choosing a method, in that case, it's because nobody wants to put soap in their mouths. That's why, okay? But so choosing a method is going to have a lot of different reasons and questions that have to be answered. And so choosing a method of control is going to be decided on by many, many, many factors. Some of the major ones are the intended use of an object, okay? So when you're looking at the intended use of an object, generally you want to think about how this object is going to be used. And would you be okay with if I took the carpet in your bedroom and turned it into a fork that you were going to be sticking into your mouth? No. I'm guessing the carpet in your bedroom is not as clean as the silverware in your kitchen, right? So we clean dishes typically every time we use them. The reason for that is because they directly go into our mouths, which is a high area for possible pathogen exposure. A lot of pathogens get in through our mouths and nose. Okay, and so your carpet is not going to have to be as clean as your silverware because you're walking on your carpet. Your silverware is going into your mouth. Now, your silverware doesn't need to be as clean as something that is going to penetrate your skin. Okay, you never want to you wouldn't want to pierce your ears with the fork in your kitchen. That's not clean enough. It's clean enough to go in your mouth, not clean enough to make a hole in your skin. Right? So the intended object is going to determine how clean something needs to be. Surgical instruments have to be cleaner than silverware. Silverware has to be cleaner than floors. Okay? And that should kind of make logical sense. Things that are going to be penetrating your body in any way, breaking the skin especially, should be more clean than something that's not. Okay? And then it also is going to depend on the type of surface. Right? Carpet is a fabric. It's going to absorb liquids and things like that. And it's very large, right? I can't boil the carpet in my house. right? That's ridiculous. Like, you say that out loud and that's ridiculous. I can't cook my carpet because it's made out of plastic. It's a fabric. It would melt, right? So all of these things, the type of surface is going to determine or dictate what type of decontamination we use, right? Like, you can use bleach on some types of clothes and fabrics, but you can't use it on others because it will ruin them. Again, I can't stick my whole mattress in my oven to try to clean it, right? That's silly. So the size of the object, the fabric of the object, is going to depend on what we choose to clean it with. And again, a lot of this should be kind of intuitive. Like, when I say that I can't dishwash my carpet, right, that sounds ridiculous because it is. And then the other factor, or another factor, are the likely pathogens. There's a reason that bathrooms, at least in this day and age, there was a time 50 or so years ago where it was really popular, but now most people don't like carpets in their bathrooms. There's a reason for that, because in your bathroom there's a lot more bacteria, because that is where you defecate, right? That is where the bacteria inside of your colon have access to the outside world, is inside of your bathroom. So there's a reason that people clean their toilets and their bathroom sinks and their bathroom floors much more often than they vacuum the rug in their bedroom, okay? Because there are likely more pathogens in the bathroom. And there's a reason why hospitals have to be cleaned more than your house, because hospitals are full of sick people. And so the number of pathogens are more likely. And then the last method that we'll talk more about in a second is the pathogen rate of resistance. So how easily the pathogens that we suspect are capable of being killed. Okay, so some microbes are more or less resistant to our current methods of microbial control. Obviously, these endospores are at the very top of the most resistant. They are the hardest to kill. Prions, which we'll talk more about later, but I wouldn't worry about right now, are the most resistant. But those endospores are incredibly, incredibly, incredibly resistant to most methods that we have for control. So in a situation where we expect an organism capable of producing endospores, we take extra precautions. There's particularly a bacteria, it's known as Clostridium difficile, produces an infection called C. diff, which we'll talk more about later. And if you do become a nurse working in a hospital, you'll hear about C. diff all the time. C. diff is a particularly gnarly and particularly vicious gastrointestinal infection. It causes just unimaginable diarrhea that is uncontrollable for weeks at a time. Okay, it's awful. And it is caused by a Clostridium that is capable of producing endospores. If somebody in the hospital, I used to work at St. Francis, if somebody in the hospital tests positive for C. diff, their room is put on complete lockdown. To go in or out, you have to have full PPE, face mask, gloves, even the cleaning staff has to be fully suited up. Visitors are limited, people are not really allowed in and out. Anything that goes into that room has to be sterilized before it comes out, even things like food trays, right? The food trays cannot be removed and taken down to the kitchen because those endospores will spread. If that patient gets an endospore on their coffee mug, they put that back on the tray, and then that goes down to the kitchen where somebody picks it up, that endospore has now spread, and it's likely to pass to somebody else because it will not be killed by the dishwasher or the hand sanitizer or anything like that. So when you suspect a high resistance pathogen like bacterial endospores, you have to modify or change your chosen method. Okay, and things that are more resistant are going to require more procedures. There are, I don't know where exactly it falls, but we'll just kind of talk about it in general. So most viruses are actually pretty susceptible to most cleaning agents, soap, hand sanitizer, things like that, will kill most viruses. They're usually pretty sensitive to our current methods of control. There is one example of a virus that probably most of you have come in contact with that is not. It's called norovirus. It also causes a pretty gnarly case of gastrointestinal distress. And it is spread very rapidly through mostly children, because again, children are gross. But it spreads really quickly because it is not killed by hand sanitizer. It's pretty resistant to bleach. It's not killed by Lysol. So any of those typical methods of control that you would use like within your own house, right? Hand sanitizing, Lysol wipes, right? The Clorox Lysol wipes, none of those work. You have to get special hydrogen peroxide bleach combo wipes that specifically say that they kill norovirus. Hand sanitizer is not going to do anything for you. You have to excessively wash your hands. You have to like sterilize or sanitize your laundry. So there's a reason why if you've ever had a holiday or been in your family and one person gets the stomach flu and then 2 days later it's ripped through your entire household, you most likely had norovirus because it spreads incredibly quickly. Because all that normal stuff where, oh, you separate them and you hand sanitize, whatever, that doesn't work. Norovirus is gonna get past all that. Okay, so again, how resistant you expect your particular pathogen to be is going to change your method of control. Like even within your own house, if my son came home and I suspected he had norovirus, I would immediately be out buying the special wipes. I would have everybody hand washing, right? The normal procedures of, oh, just sanitize my hands after I touch him and stuff like that are not good enough. I need to change my method based on the rate of pathogen resistance. Okay. And then when we talk about microbial agents, and these are things that you're going to have to know about each of our microbial agents. We're going to talk about each one. In general, there are two categories. So a type of cidal agent are agents that actively kill living bacteria. Okay? And this is like the word homicidal, right? Homicidal means you killed somebody, or homicide means you killed somebody. A cidal agent is going to actively kill bacteria. So a bactericide or a bactericidal agent is an agent that kills bacteria. A fungicidal agent is an agent that kills fungi. A virucidal agent is one that kills viruses. It kills them. Okay? A static agent inhibits and slows the growth. So a bacteriostatic agent is going to slow bacterial growth or discourage bacterial growth. So we'll give some examples later, but one of the ones that we'll talk about is the difference between heating and cooling our food. So if I cook my food, I have disinfected it. Most cooking methods kill almost everything that's living. Boiling, frying, baking, it's gonna kill just about everything that is in there. However, so heat is bactericidal, fungicidal, virucidal, all right? But putting something in the fridge is not actually gonna kill it, right? And this should be pretty evident for every time you've ever put a loaf of bread in the fridge and it's growing mold 3 weeks later, okay? The fridge is bacteriostatic. It slows down the growth because bacteria don't tend to like to grow in cold temperatures. It slows down the growth of bacteria. It slows down the growth of viruses. It slows down the growth of funguses, but it does not kill them. Okay, and so a lot of the agents that we use are actually only bacterio or fungistatic, and we're going to talk about each of them as we go through it. Okay, and this is the last slide we'll cover today, but try to avoid packing up, because once you all start packing up, it gets loud, and we have a couple more minutes. So the last thing we'll talk about before we go through particular things are the modes of action. And the mode of action of any type of control is going to be how the individual agents control the microbial growth. So what is it that they're affecting? And most of them will fall into one of the 4 general categories. and it involves affecting one of the four major parts of being a cell. So the cell wall, the cell membrane, the DNA, or the proteins. So each of these methods of control is going to be doing so because it's controlling one of these four major parts of being a cell, right? And that can happen in a lot of different ways, right? When it goes to the cell wall, you can some methods physically poke holes in the cell wall, some prevent new cell walls from being synthesized, and all kinds of stuff. But we're not gonna go into too much detail, but they will fall into one of these general four categories. They're going to affect one of the four major parts of being a cell in some way. Okay, but a mode of action is just how the individual agents control the growth. All right, that's where we will leave it today, and where we will pick up next time.